So now we know how insulin has secreted from the beta cells of the pancreas. Now blood is full of the insulin molecules and a lot of nutrients, glucose, amino acids, that already we know. So now what is the role of liver in that fed state? Exactly what is happening? So these all molecules which are present in the blood, which are going to liver through the portal vein. It is very important. Liver is present between the GIT and as well as the systemic circulation. Understand? So liver is present. So liver is acting mainly here, acting as defense against the hyperglycemia in the well-fed state. How it is? How it is? Now we are going to discuss about that. We know lot of nutrients are coming and stimulating the beta cells of the pancreas. Beta cells of the pancreas, it is releasing the insulin molecule. Lot of insulin molecules are increasing in the blood. Example, this is the blood and it is going to the liver. This is the hepatic portal vein. Hepatic portal vein, because it has to go the hepatic portal vein. And one more thing I forgot to say, during the secretion of insulin, not only insulin molecules are secreting from beta cells, C peptide is also co-secreted along with the insulin. Understand what is this C peptide? Trust me, for a while I will discuss when we come to subsequent chapters. But along with the insulin, C peptide molecules are also releasing with the insulin. So everything now it is going to the hepatocytes. What is hepatocytes? In lot of glucose is coming into the hepatocytes. What will happen then? Hepatocytes, see. So remember, liver contain one more low affinity glucose transporter, that we know. This is again glucose transporter 2, GLUT2. This is also glut GLUT2, GLUT2, GLUT2. So lot of glucose is coming into the liver. So what will liver see? I am telling very important point regarding the liver. In our body, liver is like a mother for us. Understand why? Liver is such an organ, it is selfless organ. Whenever that blood glucose glucose in our blood rises in fed state, what will happen? Liver extracts the glucose from the circulation and it is converting into the glycogen for future purpose when we are going to fasting. How does it, how that livers know? Liver has the capability again to extract the glucose from the circulation. And now lot of glucose, this is one hepatocytes example I am telling. Millions of hepatocytes are present in one liver. That of course we have only one liver. See, now glucose came. Now glucose came. Already we know, even so, this is the low affinity transporter glucose, and this glucose is converted into glucose 6 phosphate by very important glucokinase enzyme. Even glucokinase is waiting here also, because it is the time to extract the glucose. Understand what will happen? This glucose 6 phosphate ultimately going to glycolysis and generates the pyruvic acid, and this pyruvic acid going to form acetyl coenzyme A, and these acetyl coenzyme A runs Krebs cycle and involvement of ETC, and leading to the formation of abundant levels of ATP. So ATP levels are again increasing in the hepatocytes. Lot of energy is there. Like what will liver see? Like keep on coming. Glucose concentration is very high in the extracellular compartment. So increasing in ATP levels. That, but keep in mind student, in liver this is the specialized organ. It has the capability to store the glucose in the form of glycogen by a process called glycogenesis. This is very important, because we are storing food when we have abundant food. Understand, this is very important. Now Can you appreciate what is the, so lot of ATP, so then lot of ATP will tell the liver already we have enough energy, why we have to run the this oxidation pathway, better you store the glucose in the form of glycogen for future purpose. Understand? So this glucose 6 phosphate has another fate that is called glycogen synthesis. This is very important, glycogen synthesis, because this is the starting substrate for the glycogen synthesis. Okay, glycogen synthesis. And these glucose 6 phosphate can enter into one more pathway that is called HMP shunt. HMP shunt. Understand? Of course, by going to this pathway, it will gives NADPH. NADPH. I will tell you what is the role of NADPH. Even lot of glucose still it is coming. Already liver is full of energy. Lot of ATP is there. So it is keep on extracting some this glucose 6 phosphate and entering into the glycogen synthesis, and some molecules of glucose 6 phosphate will enter into the HMP shunt. It is giving the NADPH. But like so much of glucose still it is coming because we are in fed state, lot of we are eating and keep on absorbing, our organs are doing their work, keep on absorbing into the lot of insulin is secreting under the influence of insulin. Understand? Liver has the abundant insulin receptors. Understand? Insulin will stimulate the hepatocytes. You please synthesize more and more glucose in the form of glycogen. I will stimulate you, don't worry, I will tell how to store the glycogen. Understand? This is very important. So keep in mind lot of ATP is there. So these acetyl from acetyl coenzyme, lot of acetyl coenzymes are also keep on forming in the liver. What will happen then? What will happen? Yes, these, so here this acetyl coenzyme A has one more fate, because a lot of acetyl coenzymes are being formed in the hepatocytes. What will happen? So these acetyl coenzyme is entering into the fatty acid synthesis. Very important it is, fatty acid synthesis. What is the role? Because I will sum up later. See, fatty acid synthesis, it will combining with the glycerol molecule plus glycerol molecule forming the triacyl glycerol. Very important. This is the endogenous triacyl glycerol which is formed in the liver when we are in the well fed state. Understand? This is very important. Now these are the endogenously synthesized triacyl glycerol. Now in the blood, certain lipoproteins are waiting for catching these triacyl glycerols to carry into the adipose tissue where we are storing the fat. After converting these fatty acids to the triacyl glycerol, we are going to store in the adipose tissue. You know, our skinny under the skin we have a subcutaneous tissue that is called fat tissue, adipose tissue. It is consisting of again millions of adipocytes. Adipose tissue is widely distributed throughout the body. I will tell what is the role of adipose tissue again in fed state, but keep in mind what will happen. There is a one lipoprotein called that is VLDL, very low density lipoprotein. So these endogenously synthesized tag triacyl glycerol will catched by this VLDL, and ultimately it will, this is called what is that lipogenesis. We are storing in the form of triacyl glycerol in the adipose tissue. Understand? This is very important. Again I am going to sum up this. You know, not only glucose molecules are coming into liver. See what is the fate of this glucose in the liver. Now glucose is going into the oxidation pathway. It runs glycolysis, Krebs cycle and electron transport chain. It is giving lot of abundant ATP. Already our hepatocytes are rich in ATP. Now what is the fate of glucose? Yes, glucose can undergo into, can go to glycogenesis. It can go to HMP shunt. So I will be telling what is the importance. Keep in mind, for synthesis of fatty acids we need NADPH. Yes, very important it is. So for synthesizing of the fatty acids we require the this NADPH. We can use. That's why what is the purpose of HMP shunt here? Understand, this is very easy. If even if you don't understand, don't worry, I will be teaching the same concepts again. It will be repeated in the when we come to the metabolism part. Okay, so this is the role of liver in fed state. So that's why extra carbohydrates are converting into the fat. That's why like now we used to discuss with our families and everyone, you know, excess carb taking carbohydrate diet is also leading to the obese, like that we like use it to hear, right? So this is exactly what is happening in the, so amino acids are also entering into the liver. What is the purpose of amino acid? Yes, of course, formation of the proteins. It may help, it will helpful in the formation of certain polypeptides, and it can undergo, amino acids can undergo the oxidative deamination. Not only, then remaining carbon skeletons will be used in the anabolic fat. They can synthesize many compounds, then I will be telling in the subsequent chapters. So this is the role of liver in fat state. Again I am telling you, liver is like a mother, understand? So when we have excess glucose in our blood, liver has the capability to extract that glucose and converting into the glycogen. This is called glycogenesis, and certain molecules of glucose entering into the HMP shunt to yields NADPH. This NADPH again is important whenever this acetyl coenzyme, excess acetyl coenzyme will converting into the fatty acids, we require the NADPH. I will be telling in the biosynthesis of fatty acids. Now finally we formed the triacyl, endogenously synthesized triacyl glycerol we have made in the hepatocyte. Now in blood, which protein is waiting for catching this? Yes, VLDL, very low density lipoprotein. So finally this VLDL is carrying into the adipose tissue, and there we can store the TAG in the adipose tissue. Understand? So this is the role of liver in fed state. Exactly, understand. This is, if you understand, we will go to the exactly what is happening, how adipose tissue is response to the this in fed state, how what is happening. Always remember, oh, so I told you this is low affinity transfer. What does it mean? Well, GLUT2, even beta pancreas cells also has the GLUT2. What does it mean? Low affinity transporter. When the blood glucose level is rising, understand, then only it has the capability to take the glucose from the blood. What does it mean? Of course, low affinity. If the glucose concentration is low in the blood, it has low affinity, it can't take the glucose from the blood. That is why it is called low affinity and high Km. We will be studying in the enzymology. That is not a problem. So now this is the role. So one more thing I want to say, what is the role of these insulin molecule, I will be telling. So now our blood is full of glucose, we have to control the glucose. Certain tissues in our body, understand, certain glucose transporters in specific tissues are insulin dependent. What are those? See, skeletal muscle and heart and adipose tissue contain the glucose transporter 4. This is insulin dependent. What does it mean? For stimulating these glucose transporter, insulin is required. Then only these gluts will be and coming to the surface of that particular cells. I created a mnemonic which is called HAS. HAS. This is the glut for glucose dependent, sorry, insulin dependent glucose transporter. Heart, adipose tissue and skeletal muscle contain the glucose transporter 4. What will happen? Insulin is also coming and insulin is going to adipose tissue, heart and skeletal muscle and stimulating the cells. You please show your glut force. So I came, because I came, then these glucose transporters are, you know, coming to that surface and keep on starting extracting the glucose from the blood. And so that is the importance of insulin, how insulin is acting and telling to liver, you extract the glucose and store in the form of glycogen. Again telling to this heart, adipose tissue and skeletal muscle, you extract the glucose. This is the time to extract glucose because we are in well fed state now. Understand? So these adipose tissue, skeletal muscle, heart is taking glucose from the blood. Now we will see the what is the role of adipose tissue in the fed state. This is very important again. So now we will be telling the adipose tissue in the fed state. Easy. Now exactly what is happening, we will draw a adipose tissue first. Okay, so glucose enters into adipocytes by GLUT4. I told you what is the importance of this GLUT. For stimulating, for working of these GLUT, insulin is required. That is what we have studied. This is very important. If you understand this concept, we are going to deal with the diabetes mellitus. You know one fact, every second in this world, one person is dying due to the complications of the diabetes mellitus. As a good doctors, we have to know exactly what is happening at the molecular level. Understand, this is very important. Now simply I am drawing one adipocytes for our understanding. Of course, millions and millions of adipocytes are present in our adipose tissue. Understand, again this is very simple. See, suppose this is our one adipocyte. Understand, of course this is fat cell, right? Right. Again, what glucose transporter is present in the adipose tissues, my friends? Again, this is GLUT4. Always remember, this is very, very important. This is very, very important. GLUT4. So a lot of glucose is coming in the presence of insulin. Always remember, these are also low affinity or high affinity. These are also low affinity. Whenever the blood glucose levels rises, then only it has the capability to extract the glucose from the blood. That is what I told you. Yes. So again the same funda, even our body is made up of the 30 trillions of cell. Every cell needs energy. Understand? What does it mean? So glucose is entering into every cell. Always remember, whenever the glucose is entering into our cells, so always enzymes are ready to take that glucose, adding phosphate to glucose at 6th position. This is what exactly happening at our molecular level. So this glucose is converting into glucose 6-phosphate by here hexokinase. Of course, I will be telling the differences between these hexokinase and glucokinase, but keep in mind this is. So again we know the same funda. This glucose 6-phosphate will entering into the glycolysis, forming the pyruvic acid. This pyruvic acid will go to pyruvic acid for converting into acetyl coenzyme A. This acetyl coenzyme A is converting into like it runs the TCA cycle, and finally electron transport sign, again leading to increase in the synthesis of the ATP. This is very, very important. Understand? Always remember, of course even adipocytes needs energy, right? So here you have to understand, when the ATP levels are increasing in the same adipocyte tissue, so here we have to understand these acetyl coenzyme A, this acetyl coenzyme is converting into the fatty acids. Understand, this is very important. Fatty acid synthesis, it is entering into fatty acid synthesis. Always remember, we told you, again we are adding the glycerol molecule. This glycerol molecule, whenever I am adding glycerol to the fatty acids, what it will form? Triacyl glycerol. This triacyl glycerol it is also endogenously synthesized by the adipocytes, within the adipocytes in the well-fed state. Always remember, it is occurring only in the well-fed state. When we have the abundant glucose levels, when the glucose level spike increases in the blood, this is very, very important. So this triacyl glycerol will store it in the same adipocyte. Did you remember, as I said earlier, in livers something we have studied, some tag endogenously synthesized tag is converting into the this, like catched by this VLDL. Of course, from this VLDL from the liver, very important, this VLDL carries the what fat which is synthesized by the liver. Example, from liver VLDL is coming, and from our diet, whatever the diet we are taking, the triacyl glycerols in the diets which is carried by the chylomicrons. It is very important. Chylomicron. This is exogenous triacyl glycerol. This is diet. So this chylomicron, what it will happen? Both will give same. What it will give? Triacyl glycerol, and this tag. So it will give in the form of fatty acids. Of course, this will split into fatty acids plus glycerol. It will give. ఇక్కడ కట్ చేయండి తర్వాత, ఓకే, అదేలే, రైట్. Here VLDL and chylomicrons, VLDL from liver and chylomicrons from the diet, giving the tag. So ultimately here it is splitting into fatty acids plus glycerol. Again, this fatty acid adding a glycerol molecule, and we are storing the triacyl glycerol in the same adipose tissue. What will happen in adipose tissue? We are synthesizing the triacyl glycerol. So this is all adipocyte. If you look at the adipocyte, how it will be, so small lobular, small nucleus, and remaining, these are all fat only. The whole cytoplasm is filled with the fat, because we are synthesizing in the adipose tissue. We are taking the tag from liver, we are taking the tag from the diet. So this is the role of adipose tissue in the fed state. This is very interesting. Of course, one thing I remember, this glucose 6-phosphate has it should undergo the HMP shunt. Can you tell me why it should undergo the HMP shunt? Because it is giving the NADPH. I told you NADPH is used in the biosynthesis of fatty acids in our body. Understand? That's why some glucose molecules will entering into alternate pathway, that is called HMP shunt. So again, I am repeating the adipocyte. What will happen? I am taking lot of food, and adipose tissue, in the presence of insulin is going to stimulate the adipose tissue. Glut force will be coming into the surface of the adipose tissue. This will get stimulated and keep on absorbing lot of glucose from the blood. Understand? This is very important. So lot of glucose, rush of glucose coming into adipose tissue, which is taken by the glucose transporter 4, which is by the process of facilitated diffusion. Because you can understand, this is the extracellular is rich in glucose, so concentration gradient from high concentration to low concentration. Right, in well-fed state, exactly what is happening in our skeletal muscle? I told you insulin dependent glucose transporters are present in the hot adipose tissue and skeletal muscle. Now we are coming to the skeletal muscle. So I am drawing one skeletal muscle here. I am not good at diagrams, but you please see what I am drawing, that's it. Okay, suppose this is the skeletal muscle. Example, this is skeletal muscle. This is very big skeletal muscle. Okay, this is skeletal muscle. Exactly what is happening in skeletal muscle? Already we know what is the glucose transporter present in the skeletal muscle. Of course, this is also GLUT4 only. So lot of glucose is coming with the help of insulin is telling to this glucose transporter, take the glucose now, it is the time to get the glucose, otherwise you won't get later, because you have GLUT4. This is insulin dependent. Understand? So lot of glucose is coming. This glucose is converting into again glucose 6-phosphate by here important enzyme. We know that is glucose is converting into glucose 6-phosphate, and glucose 6-phosphate is runs the glycolysis, giving acetyl coenzyme A, and acetyl coenzyme A leading into the entering into TCA cycle, and ETC gives the lot of ATP. Finally, skeletal muscle also fulfill their dreams, but always remember, skeletal muscle play very important role in getting the glucose to control the blood glucose. So pyruvate, acetyl coenzyme A, again TCA cycle. These we know. Super. Always remember, in adipose tissue we didn't mention the glycogenesis, but here I have to mention this glucose After like getting enough energy, what will happen? Muscle has the capability to run the machinery of glycogenesis. So it is synthesizing the glycogen in the fed state in the influence of the insulin. Understand, this is very important. Of course, not only glucose is entering into my muscle cells, lot of nutrients are entering, even amino acids are abundant in the blood, which are keep on taking by the muscles for the synthesis of the proteins. And proteins are the structural properties of our muscle. There we have so many muscles in our body. So this is what exactly happening in the skeletal muscle. So now we are coming to very important thing, that is our hero of our homeostatic balance, that is brain. Exactly what is happening in this brain? What is happening in this brain? Always remember, brain has very important glucose transporter, that is GLUT1 and 3. GLUT1 and 3, these are not insulin dependent. Brain never listen any words, it will do whatever it wants. Understand, always remember, brain always dependent on the glucose only. It never take fatty acids or some, because blood brain barrier, glucose is freely cross this blood brain barrier through these GLUT1 and GLUT3. Very important, brain always dependent upon glucose in the well fed state, of course in fasting state also, but at certain stages it will depend upon the ketone bodies. I will be teaching later. So there are two glucose transporter 1 and glucose transporter 3. One is present at the blood brain barrier, 3 is present at the neuronal level. Understand, this is very important. Always remember, both are high affinity transporters, both are high affinity transporters. What does it mean? Even if the even if the glucose concentration in the blood is very low, but it has the capability to take the glucose from the blood. That is why it is called high affinity towards the glucose. Even blood glucose concentration is less, but as we said earlier, GLUT2 is low affinity, GLUT4 is low affinity. So what does it mean? Low, don't confuse between low affinity and high affinity. High affinity in the sense, even if the blood glucose level is very less, but it has the capability to extract that minimal basal uptake of glucose. This is very important to understand the brain. Transport of glucose occurs into the brain in all stages. I can say again, this transport of glucose into the brain occurs in all the stages, even if it is fasting, fed state, this is whatever glucose is present, it will be absorbed by our brain. It is very important. Again, so this is very important. Suppose again, glucose is converting into, glucose is coming into the neurons by the GLUT1 and 3. So it is converting into glucose 6 phosphate. We know this glucose 6 phosphate is converting into the pyruvic acid. This pyruvic acid is converting into the acetyl coenzyme. Same story. This is the biological mechanisms which are occurring in every cell, of course except RBC, you should know, which runs only anaerobic glycolysis because it lacks the mitochondria and nucleus as well. Understand, that's why where our body exceptions are RBCs only, RBCs and the skeletal muscle in case of exercise. Understand, they will switch to the anaerobic glycolysis. I will be telling in the glycolysis chapter. This is the just overview. Even if you don't understand, no problem at all. So these acetyl coenzyme A gives a lot of energy by running the Krebs cycle or TCA cycle and increasing in the ATP molecules. This we have understood, because this is exactly, we contain the billions of neurons. See how God has created our body in such organized manner. We can't imagine also, our body is made up of more than 30 trillions of cells. So exactly what is happening, we are taking the diet in millions of glucose molecule. Every cell needs energy. Understand, every cell will run this biochemical pathway to yield the ATP. This is very important. Now we are going to wind up this biochemistry of fed state with very important table important table. So example, this is very important student as per our exams also, which is very important. This is for organ and this is the metabolic fuel. Always remember, this is metabolic fuels. First brain, RBCs, liver, heart, adipose tissue or adipocytes, skeletal muscle. Can you appreciate HA as glucose transporter 4, which is insulin dependent? Skeletal muscle, this is okay. So tell me, now you have to tell what this is the organ, this is the fuel. What brain will use? Always remember, in fed state, brain only depends upon the glucose 100%. Keep in mind. If you see liver, of course, RBC is also only depends upon the glucose in all stages. It never depends on any other sources, because the pores of RBC doesn't contain mitochondria. That we know from 10th class we are studying. So liver, yes, in fed state, we are in fed state, glucose only, abundant glucose we have. So glucose greater than, it can use fatty acids to run more accurately. Understand, this is very important. Heart, always remember, of course we know, but remember, exception in the fed state, this is very important. Exception in the fed state, even we have the abundant source of the glucose, heart will use free fatty acids. Why? I will tell later. Understand, free fatty acid, then glucose. I will tell, because our heart have to beat all the time to pump the blood into circulatory system. 1 lakh beats per day. How does it? It requires more energy. Understand? And one more thing, from different sources, it has the low glycolytic capacity. Low glycolytic capacity. Understand? I will tell when we come to the particular metabolisms, then you will understand. Like when we come to glycolysis, I will teach you. In adipose tissue, of course, if we are in fat state, it will absolutely will use glucose only. Glucose greater than free fatty acids. If you see the skeletal muscle, of course glucose only. This is very important. Examiner can ask these kind of questions also. When I was in first year, I was so confusing, what is this cell, like that. But once I started reading biochemistry is such an interesting subject. What is exactly happening in our body, so what kind of things are happening in our body, like that. I am so fascinated to teach this biochem subject. So glucose, then free fatty acids. So this is our biochemistry of fat state, what we have studied. So this is the metabolic fuels we have studied till now. So I am going to wind up the class of biochemistry of fat state, students. Now just I am giving the whole review of total what we have studied till now. See, I am taking food which is rich in carb, protein and lipids. It is going to my GIT. GIT will have, GIT knows, GIT contains some neuroendocrine cells. Yes, nutrients are coming. So then GIT will secrete the lot of incretin molecule, and these GIT, after digestion of foods, after coming these macromolecules to micromolecules, it is absorbing along with the all nutrients, so glucose, free fatty acids, amino acids and vitamins and minerals, all it will go to the blood. So whenever these are going to blood, it will go to pancreas and telling to pancreas, you please secrete insulin, because that spike of glucose is going to come, lot of glucose is coming, and incretins will tell the beta cells of pancreas to amplify to. Already you are secreting insulin, but it is not enough. You have to secrete more insulin. Later, lot of food still I am eating, so still food is nutrients are coming, glucose is coming, so it will amplify the insulin release, and the insulin is co-secreted with the C-peptide to the blood, and it will go to the liver to the portal vein. And what will liver do? Liver is like a mother. Liver is extracting the extra glucose, lot of glucose it is keep on absorbing and runs the machineries that is called glycogenesis, HMP shunt, and access acetyl coenzyme from the pyruvic acid. And what will happen? These acetyl coenzyme, the fat is here to convert the fatty acids. These fatty acids will combine with the glycerol molecule to form the endogenously synthesized triacyl glycerol. So it will taken up by the VLDL, which is present in the blood as the lipoprotein, and it will take that particular TAG which is synthesized by the liver and going to the adipocytes to store in the fat tissue. This is what we have studied. And how the skeletal muscle role play, because there are some glucose transporters which are insulin dependent, they will come only when insulin is there, understand? So when insulin will come, of course, whenever the blood glucose level, when the thing is that when insulin starts secrete in our blood, yes, when the blood glucose level increases, when blood glucose level started increasing more than 70 milligram per deciliter. This is very important. So insulin is coming, insulin is telling the heart, adipose tissue and skeletal muscle, glucose transporter 4 will come, and a lot of glucose is keep on absorbing. In that way we are decreasing the glucose level in the blood. And hepatocytes are also, what the hepatocytes will do, and by stimulation of the insulin, it will undergo the process of the glycogenesis and HMP shunt and access acetyl coenzyme converting into the fatty acids, and which will be taken up by the VLDL that we know. So again, heart. So same, we have studied what is exactly happening in the RBC. RBCs always takes glucose in fed state, whether fasting state, whatever state it is. So this is the exactly what is happening in our body when we have taken a lot of food. These are the biochemical reactions which are, this is just overview. From next chapter, that is the biochemistry of fasting state, I will be telling exactly what is happening if you are not taking food for long hours. Understand? If you understand, and this is my first video, that's why look, if any mistakes are there, you please forgive me guys, and you please comment in the chat box how to improve myself to teach in more accurately. I want to share my all biochem knowledge with you all people, but this is first video, I don't know how it came also, but you please support. And even if you don't like, I will improve myself for how to teach. Please teach me how to teach. Please give a chance to complicated topics to simple manner. Please help me. Again, if any mistakes are there, please forgive me, and give more channel and give more chances to improve myself. And please subscribe my channel, and I will get more motivation to do lectures with more enthusiasm. Thank you so much guys, and next video we will discuss about the biochemistry of fasting state. Thank you so much, and subscribe Doctor's Care channel.